Why is it necessary to fix a biological specimen?
Fixing a biological specimen retains a reasonably good semblance of the object as it appeared when it was alive. It allows the scientists to observe details of the external and internal anatomy of the specimen.
What is electrophoresis?
Electrophoresis is a technique used to separate biological molecules, such as nucleic acids, carbohydrates, and amino acids, based on their movement due to the influence 532 of a direct electric current in a buffered solution. Positively charged molecules move toward the negative electrode, while negatively charged molecules move toward the positive electrode.
Do animal studies predict human behavior?
George Romanes, 1848 to 1894. Was one of the first scientists to investigate the comparative psychology of intelligence. He believed that by studying animal behavior, one could gain insights into human behavior. However, his theory was based on inferences rather than direct observations of comparable behavior.
How do dissecting microscopes differ from compound microscopes?
Compared to compound microscopes, dissecting microscopes, also called stereoscopic microscopes, provide a much larger working distance between the lens and stage in order to dissect and manipulate specimens. The light source on a dissecting microscope is above the specimen, since the specimen is often too thick to allow light to be transmitted from a light source below the specimen. Dissecting microscopes are always binocular, which provides a three-dimensional image.
What main carbohydrate is stored by fungi?
The main carbohydrate stored by fungi is glycogen, which is also the main storage carbohydrate of animals. This fact suggests that fungi are more closely related to animals than plants, which store starch as their main carbohydrate.
Do animals ever help each other?
Populations of organisms within an environment may engage in a variety of relationships with each other. For example, in a relationship known as mutualism, each species provides a benefit to the other. Mutualism can occur between two animal species, like the relationship between large coral reef fishes and the smaller species, like the wrasses that swim into their mouths and eat the parasites that may have taken up residence there. Mutualistic relationships also allow bean plants to grow better because they exchange nutrients with rhizobium fungi living on their roots.
What are vitamins and minerals?
A vitamin is an organic, non-protein substance that is required by an organism for normal metabolic function but cannot be synthesized by that organism. In other words, vitamins are crucial molecules that must be acquired from outside sources. While most vitamins are present in food, vitamin D, for example, is produced as a precursor in our skin and converted to the active form by sunlight. Minerals, such as calcium and iron, are inorganic substances that also enhance cell metabolism. Vitamins may be fat or water-soluble. Recommended amounts of vitamins are to ensure normal enzymatic function, and excessive intake can be toxic.
What are giant tube worms?
These worms were discovered near the hydrothermal, hot water ocean vents in 1977 when the submersible Alvin was exploring the ocean floor of the Galapagos Ridge. Located 1.5 miles 2.4 kilometers below the Pacific Ocean surface and 200 miles 322 kilometers from the Galapagos Islands. Growing to lengths of 5 feet 1.5 meters, Riptia pachyptila Jones. Named after worm expert Meredith Jones of the Smithsonian Museum of Natural History. Lack both mouth and gut and are topped with feathery plumes composed of over 200,000 tiny tentacles. The phenomenal growth of these worms is due to their internal food source symbiotic bacteria. Over 100 billion per ounce of tissue, that live within the worm's troposome tissues. To these troposome tissues, the tube worms transport absorbed oxygen from the water, together with carbon dioxide and hydrogen sulfide. Utilizing this supply, the bacteria in turn produce carbohydrates and proteins that the worms need to thrive.
What is a pseudogene?
As genes are copied and transposed, mutations may occur that make the copies non-functional. This is thought to be the origin of pseudogenes, which resemble actual genes but which, while stable, do not produce a polypeptide.
Are freshwater clams an endangered group?
Although freshwater clams are found on every continent except Antarctica, they are now considered one of the most jeopardized groups of animals in the world. Approximately 270 species belong to the family Unionidae, found in North America. A total of 72% of our 270 native mussel species are listed as recently extinct. endangered, threatened, or of special concern due to human impact on aquatic habitat, commercial harvesting. The introduction of carp, water pollution, and the invasion of zebra mussels.
What is the most widely cultivated cereal in the world?
Wheat is the most widely cultivated cereal in the world. The grain supplies a major percentage of the nutrients needed by the world's population. Wheat is one of the oldest domesticated plants, and it has been argued that it laid the foundation for Western civilization. Domesticated wheat had its origins in the Near East at least 9,000 years ago. Wheat grows best in temperate grassland biomes that receive 12 to 36 in 30 to 90 centimeters of rain per year and have relatively cool temperatures. Some of the top wheat-producing countries are Argentina, Canada, China, India, the Ukraine, and the United States.
How are nitrogenous wastes metabolized?
Ammonia, urea, and uric acid are nitrogenous waste products. They are the result of the breakdown of various molecules including nucleic acids and amino acids. Some amino acids are used for new protein synthesis, while others are oxidized to generate energy or converted to fats or carbohydrates that can be stored. Once broken down, the amino group NH2, containing 1 nitrogen and 2 hydrogens, must be removed or the animal will eventually be poisoned. Ammonia is the most toxic of the nitrogenous wastes. It is formed by the addition of a third hydrogen atom to the NH2 group. Both urea and uric acid are less toxic forms of nitrogenous waste but have greater energy requirements for their production.
In the home setting, does the use of antibacterial soap reduce the risk of infection?
Research has uncovered little evidence to support claims that the use of antibacterial soap in a domestic setting reduces the risk of infection or prevents infections. These residue-producing antibacterials help control the spread of infection in healthcare settings, such as hospitals and nursing homes.
What are some examples of fermented foods?
Fermented foods are foods produced by microorganisms. A combination of bacteria and fungi are used in the fermentation process of many food products. Examples of fermented foods are cheese, sour cream, yogurt, sauerkraut, vinegar, olives, pickles, soy sauce, miso, chocolate, coffee, and most alcoholic beverages.
How does water move up a tree?
Water is carried up a tree through the xylem tissue in a process called transpiration. Constant evaporation from the leaf creates a flow of water from root to shoot. The roots of a tree absorb the vast majority of water that a tree needs. The properties of cohesion and adhesion allow the water to move up a tree regardless of its height. Cohesion allows the individual water molecules to stick together in one continuous stream. Adhesion permits the water molecules to adhere to the cellulose molecules in the walls of xylem cells. When the water reaches a leaf, water is evaporated, thus allowing additional water molecules to be drawn up through the tree.
What are imperfect fungi?
Imperfect fungi are also called deuteromycetes or conidial fungi. They are an assemblage of distinct fungal species that are known to reproduce only asexually. The sexual reproductive features have not been identified and are not used as the basis for classification. In this group, sexual reproduction has not been observed. Most imperfect fungi are thought to be ascomycetes that have lost the ability to reproduce sexually. The best known members of the deuteromycetes are the genera Penicillium and Aspergillus. Whenever a mycologist discovers a sexual stage in one of these fungi, the species is reclassified from the imperfect category to a particular phylum. The phylum selected depends on the type of sexual structures.
How do yeasts differ from other fungi?
Yeasts remain unicellular, that is, as single cells throughout their life. Most species reproduce by budding, others through binary fission or spore formation. Each bud that separates from its mother yeast cell can grow into a new yeast cell. Some yeast cells group together to form colonies.
Are humans the only animals who can think?
Before one can answer this question, one must define what is meant by thought.
What is a molecule?
Molecules are made of specific combinations of atoms. For example, carbon dioxide is made of one carbon atom and two oxygen atoms. Water is made of two hydrogen atoms and one oxygen atom, and the atoms are joined by chemical bonds. Complex molecules such as starch may have hundreds of participating atoms linked in a specific pattern.
How do bacteria survive without nutrients?
When a population of bacteria loses its food supply, many bacteria dehydrate. During this process, bacteria produce a thick, tough spore coat. As spores, bacteria are able to rest for long periods of time. When conditions become favorable, the spores become active again. The bacteria absorb water, break down their thick, tough spore coats, and begin to form new cell walls.
How is the Cambrian explosion related to Evo-Devo?
Mutations in the suite of genes that control developmental processes, known as the Hox genes, would have caused the radical changes in the body structure of animals that occurred during the Cambrian explosion. These genes have been identified in every major animal group, both vertebrates and invertebrates. So it seems likely that they date back to the Precambrian period. Thus, the mechanisms that control the development of the animal embryo, Devo, also probably played a role in the evolution of animals as a group, thus the term Evo-Devo.
Can we make artificial cells?
Research in progress at the National Aeronautical and Space Administration, NASA, is focused on artificial cells as a means to deliver medicine in outer space. These cells are able to withstand dehydration and thus can be safely stored for long periods. Artificial cells are made of a polymer that acts like a cell membrane. But the polymer is stronger and more manageable than real membranes. These polymers are called polymersomes and can be made to cross-link with other polymers. Researchers feel that many different kinds of molecules can be encapsulated within these polymersomes and then delivered to specific target organs. An example would be an artificial blood cell that not only delivers oxygen but also medication as it travels through the body.
What are the shortest and longest chromosomes?
Among human chromosomes, the length ranges from 50 to 250 million base pairs. The longest chromosome is chromosome 1, with 300 million bases, approximately 10% of the human genome. And the shortest is chromosome 21, with 50 million bases.
How can animals be grouped according to type of body cavity?
The structures of most animals develop from 3 embryonic tissue layers. The outer layer, the ectoderm, gives rise to the outer covering of the body and the nervous system. The inner layer, the endoderm, forms the lining of the digestive tube and other digestive organs. The middle layer, the mesoderm, gives rise to most other body structures, including muscles, skeletal structures, and the circulatory system. One system of grouping animals with 3 germ layers, triploblastic, is based on the presence and type of body cavity. Or chylome, a fluid-filled space between the body wall and digestive tube. Achylomates such as flatworms, phylum Platyhelminthes, have a solid body with no body cavity. Pseudocoelomates have a false chylome, since the body cavity is not completely lined with mesoderm. Pseudocoelomates include such animals as roundworms, phylum Nematoda, and Rotifers, phylum Rotifera. Coelomates, which have a body cavity that is completely lined with mesoderm, include most other animals ranging from earthworms, phylum Annelida, to vertebrates, phylum Chautata.
What is the purpose of rhizoids?
Rhizoids are a characteristic feature of mosses, liverworts, and hornworts. Rhizoids are slender, usually colorless projections that consist of a single cell or a few cells. They serve to anchor mosses, liverworts, and hornworts to their substrate and absorb water.
When were cells first studied?
The scientific study of cells developed gradually from the first description of cells in the 17th century. During this early period, investigations of cells were done using the primitive light microscopes available at the time and consisted almost entirely of morphological descriptions of cell structure. In the 18th and 19th centuries, research expanded to include the study of cell chemistry and physiology. Efforts that proceeded independently from morphological studies. The study of cell structure, cell chemistry, and cell physiology continued as separate fields of experimentation until the beginning of the 20th century, when the rapidly developing field of biochemistry began to influence cell biology. At the same time, genetics became established as a field of investigation.
What is Hugo?
HUGO, also known as the Human Genome Organization, is an international consortium created to coordinate the work of human geneticists around the world.
Why is the shape of an enzyme so important?
Shape is critical to the function of all molecules, but especially enzymes, which are three-dimensional. The active site of an enzyme is the area where substrate binds and the reaction takes place. How an enzyme reacts with its substrate is similar to how a ship docks. There are minor bonds that form between the enzyme and substrate until docking is complete. Anything affecting a protein's shape would have an effect on its ability to react with the substrate.
Who is the father of mycology?
Elias Fries, 1794 to 1878, is considered the father of mycology. Born in Sweden, he received a degree in philosophy from Lund University in 1814. His first important work on mycology, Observations Mycologici, was published between 1815 and 1818. He devoted his career to the study of botany, concentrating specifically on fungi and lichens. The life cycle of a mushroom.
What is the advantage of thin layer chromatography?
Thin layer chromatography is a much quicker technique than paper chromatography. To separate a mixture using paper chromatography may take 24 hours. But less than 1 hour using thin layer chromatography.
Who is known as the founder of botany?
The ancient Greek scientist Theophrastus, ca. 372 ca. 287 BCE, is known as the father of botany. His two works on botany, on the history of plants and on the causes of plants, were so comprehensive that 800 years passed before any new significant botanical information that had not been covered by Theophrastus was discovered. He integrated the practice of agriculture into botany and established theories regarding plant growth and the analysis of plant structure. He related plants to their natural environment and identified, classified, and described 550 different plants.
Who was Mendel?
Gregor Mendel, 1822 to 1884, is the founding father of experimental genetics. His work with the garden pea, Pisum sativum, was not consistent with the 19th century ideas of inheritance. Mendel was the first to demonstrate transmission of distinct physical characteristics from generation to generation.
Who developed the gram stain and why is it important?
Staining is a process in which a scientist colors a microorganism with a dye that emphasizes certain structure. The gram stain was developed in 1884 by Hans Christian Gram, 1853 to 1938. A Danish physician who worked at a hospital in Berlin, Germany. The gram stain is one of the first steps in the identification of bacteria, and is used to categorize bacteria into two major groups, gram-positive and gram-negative. When the gram stain is combined with other information on cellular morphology and biochemical characteristics, conclusive identification of an unknown type of bacteria can usually be made. The gram stain is the most widely used stain in microbiology.
Which lichens are most often used by the perfume and cosmetic industries?
The musk-like fragrance and fixative properties of Avernia prunistri and Pseudavernia furfurnacea make them popular components for perfumes and cosmetics. The essential oils of these lichens are extracted with solvents E. prunistri and P. furfurnacea are common to southern France, Morocco, and the Serbo-Croatian peninsula.
What are the different types of natural selection?
There are several ways in which natural selection can cause a population to change. Natural selection can cause a trait to change in one direction only. For example, when individuals within a population grow taller with each generation, this is known as directional selection. Diversifying selection can cause the loss of individuals in the mid-range of a trait. For example, if there is a range in color of a certain prey species from very dark to very light, those individuals in the mid-color range may not be able to hide from predators. The mid-color prey will then be selectively removed from the population, leaving the population with only two forms, the very dark and the very light. In stabilizing selection, those at either end of the range are removed more often, creating selection pressure for the mid-range. Selection can also work on traits important to sexual reproduction. This is known as sexual selection.
What are the three common types of leaf arrangement on a stem?
The three most common types of leaf arrangement are 1. Alternate, or spiral, 2. Opposite, and 3. Whorled. In many plants the leaves are arranged in a pattern alternating on either side of the stem. In other plants the leaves are opposite each other in pairs on the stem. In still other plants the leaves are whorled with 3 or more attached to one level of the stem.
What are some commercial uses of red algae?
The cell walls of red algae contain a mucilaginous outer component usually composed of agar and carrageenan. Agar is used for making gel capsules, a material that is used to make dental impressions and as a base for cosmetics. It is also the basis of the scientific laboratory media used for growing bacteria, fungi, and other organisms. Agar is also used to prevent baked goods from drying out and for rapid setting jellies. Carrageenan is used as a stabilizer for emulsions such as paints, cosmetics, ice cream, and many dairy products. The genus Porphyra, commonly called nori, is cultivated as a food and is specifically popular in Japan.
What is the function of the nucleolus?
The nucleolus is a large, spherical structure present in the nucleus of a eukaryotic cell. It is the site where ribosome subunits are assembled, and where both ribosomal RNA synthesis and processing occur.
How do fossils form?
Fossils form rarely, since an organism is usually consumed totally or scattered by scavengers after death. If the structures remain intact, fossils can be preserved in amber, hardened tree sap. Siberian permafrost, dry caves, or rock. Rock fossils are the most common. In order to form a rock fossil, three things must happen: 1. The organism must be buried in sediment, 2. The hard structures must mineralize, and 3. The sediment surrounding the fossil must harden and become rock. Many rock fossils either erode before they can be discovered, or they remain in places inaccessible to scientists.
What are the nitrogenous bases of DNA?
The nitrogenous bases have a ring of nitrogen and carbon atoms with various functional groups attached. There are two types of nitrogenous bases. They differ in their structure. Thymine and cytosine have a single ring structure, while adenine and guanine have double ring structures. When Watson and Crick were imagining how the bases would join together, they knew that the pairing had to be such that there was always a uniform diameter for the molecule. It therefore became apparent that a double ring base must always be paired with a single ring base on the opposite strand.
Which antibiotics are most effective against gram-positive bacteria? And which are most effective against gram-negative bacteria?
Penicillin and its semi-synthetic derivatives are effective against gram-positive bacteria. The aminoglycosides, such as gentamicin, neomycin, and kanamycin, are effective against gram-negative bacteria. Antibiotics that are effective against a broad range of gram-positive and gram-negative bacteria are called broad-spectrum antibiotics.
When were bacteria discovered?
Antony van Leeuwenhoek, 1632–1723, a Dutch fabric merchant and civil servant, discovered bacteria and other microorganisms in 1674 when he looked at a drop of pond water through a glass lens. Early, single-lens instruments produced magnifications of 50 to 300 times real size. Approximately 1/3 of the magnification produced by modern light microscopes. Primitive microscopes provided a perspective into the previously unknown world of small organisms, which von Leeuwenhoek called animalcules in a letter he wrote to the Royal Society of London. Because of these early investigations, von Leeuwenhoek is considered to be the father of microbiology.
How can plastics be made biodegradable?
Plastic neither rusts nor rots. This is an advantage in its usage, but when it comes to disposal of plastic, the advantage turns into a liability. Degradable plastic has starch in it so that it can be attacked by starch-eating bacteria to eventually disintegrate the plastic into bits. Chemically degradable plastic can be broken up with a chemical solution that dissolves it. Used in surgery, biodegradable plastic stitches slowly dissolve in the body fluids. Photodegradable plastic contains chemicals that disintegrate over a period of 1 to 3 years when exposed to light. 25% of the plastic yolks used to package beverages are made from a plastic called E-collet, which is photodegradable.
How is a DNA molecule held together?
Although DNA is held together by several different kinds of chemical interactions, it is still a rather fragile molecule. The nitrogen bases that constitute the rungs of the ladder are held together by hydrogen bonds. The sides of the ladder, the phosphate and deoxyribose molecules, are held together by a type of covalent bond called a phosphodiester bond. Because part of the DNA molecule is polar, the outside of the ladder, and the rungs, nitrogen bases, are nonpolar, there are other interactions called hydrostatic interactions that occur between the hydrogen and oxygen atoms of DNA and water. The internal part of the DNA tends to repel water, while the external sugar phosphate molecules tend to attract water. This creates a kind of molecular pressure that glues the helix together.
What is a cell?
A cell is a membrane-bound unit that contains hereditary material, DNA. And cytoplasm, it is the basic structural and functional unit of life.
How many species of fungi have been identified?
Scientists have identified between 70,000 and 80,000 species of fungi, almost 2,000 new species are discovered each year. Some mycologists estimate that there are about 1.5 million species worldwide, placing the fungi second only to insects in the number of different species.
What are the most primitive animals that have been studied for their behavior?
Sponges are the most primitive animals studied for their behavior. Investigations have shown that sponges in their natural environment may move away from physical contact with competitors, areas with limited food, or sites that have excessive exposure to silt abrasion by water flows.
What is recombinant DNA?
Recombinant DNA is hybrid DNA that has been created from more than one source. An example is the splicing of human DNA into bacterial DNA, so that a human gene product is produced by a bacterial cell.
When was the term homeostasis first used?
Walter Bradford Cannon, 1871–1945, who elaborated on Claude Bernard's concept of the milieu intérieur, used the term homeostasis to describe the body's ability to maintain a relative constancy in its internal environment.
Which organelle is absent in fungi?
Centrioles, which divide and organize the spindle filters during mitosis and meiosis, are lacking in all fungi. The nuclear envelope does not break down and reform. Instead, a spindle apparatus is formed within the envelope. Fungi regulate the formation of microtubules during mitosis with small amorphous structures called spindle plaques.
Why are cells so small?
Cells come in a variety of sizes and shapes. Bacteria are among the smallest, 0.2 to 0.3 µm in diameter, while cells of plants and animals are generally larger, 10 to 50 µm in diameter. Cellular size is determined by the surface/volume ratio required to maintain access to the substances and enzymes that cells need to complete their functions. For further understanding of the surface/volume ratio, imagine that a cell is the shape of a cube. The volume of a cube increases as does the length or diameter, but the surface area only increases as the cube does. Once the upper limits of surface area/volume ratio are reached, the cell generates more cytoplasmic volume than can be met by the small increases in membrane surface area.
What are the parts of a stem?
A stem has nodes and internodes. The nodes are the points where the leaves are attached to the stem. The internodes are the parts of the stem between the nodes.
How do cells drink?
Cells drink by a process called pinocytosis, which is a form of endocytosis. During pinocytosis, the cell membrane folds inward, forming a small pocket vesicle around fluid that is directly outside the cell membrane. Fluid consumed by cells may contain small molecules, such as lipids. Endothelial cells, which line capillaries, are constantly undergoing the process of pinocytosis. Drinking from the blood within the capillaries.
What are lichens?
Lichens are organisms that grow on rocks, tree branches, and bare ground. They are composed of two different entities living together in a symbiotic relationship. 1. A population of either algal or cyanobacterial cells that are single or filamentous. And 2. Fungi. Lichens do not have roots, stems, flowers, or leaves. The fungal component of a lichen is called the mycobiont, from the Greek terms mykes, which means fungus, and bios, meaning life, and the photosynthetic component is called the photobiont. From the Greek terms photo, meaning light, and bios, meaning life. The scientific name given to the lichen is the name of the fungus and is most often an ascomycete. As the fungus has no chlorophyll, it cannot manufacture its own food, but it can absorb food from algae. Lichens and algae enjoy a symbiotic relationship. Lichens can often be found growing around and on top of algae, providing the algae with protection from the sun, thus decreasing the loss of moisture. Fungi and algae were the first organisms recognized as having a symbiotic relationship. A unique feature of this relationship is that it is so perfectly developed and balanced that the two organisms behave as a single organism.
How are genes controlled?
Genes are controlled by regulatory mechanisms that vary by whether the organism is a prokaryote or a eukaryote. Bacteria, prokaryote, genes can be regulated by DNA-binding proteins that influence the rate of transcription, or by global regulatory mechanisms that refer to an organism's response to specific environmental stimuli such as heat shock. This is particularly important in bacteria. Gene control in eukaryotes depends on a complex set of regulatory elements that turn genes off and on at specific times. Among these regulatory elements are DNA-binding proteins as well as proteins that, in turn, control the activity of the DNA-binding proteins.
What is the dikaryotic phase of fungal life cycles?
The dikaryotic phase of the fungal life cycle is unique. During this unusual phase, which is common in many species of fungi, cells. Contain two distinct nuclei. These two nuclei divide simultaneously as the mycelium grows. Growth continues until fusion occurs during karyogamy.
What is a double-blind study?
In a double-blind study, neither the subjects of the experiment nor the persons administering the experiment know the critical aspects of the experiment. This method is used to guard against both experimenter bias and placebo effects.
What is punctuated equilibrium?
Punctuated equilibrium is a model of macroevolution first detailed in 1972 by Niles Eldredge, 1942, and Stephen J. Gould, 1941 to 2002. It can be considered either a rival or supplementary model to the more gradual moving model of evolution posited by neo-Darwinism. The punctuated equilibrium model essentially asserts that most of geological history shows periods of little evolutionary change, followed by short, geologically speaking, a few million years, periods of rapid evolutionary change. Gould and Eldredge's work has been buttressed by the discovery of the Hox genes that control embryonic development. Hox genes are found in all vertebrates and many other species as well. They control the placement of body parts in the developing embryo. Relatively minor mutations in these gene sequences could result in major body changes for species in a short period of time, thereby giving rise to new forms of organisms and therefore new species.
What is pleiotropy?
Pleiotropy refers to a case where one gene may influence several other characteristics. An example of pleiotropy is sickle cell anemia, a disorder in which a single point mutation in the amino acid sequence for hemoglobin results in a spectrum of effects. Red blood cells produce abnormal hemoglobin molecules, which, because of their odd shape, tend to stick together and crystallize. Therefore, the normal disc shape of red blood cells changes to a sickle shape. Hence the name of the disorder. Sickle-shaped red blood cells will clog small vessels, causing pain and the possibility of brain damage and heart failure. Since some of the hemoglobin in these cells is abnormal, there is less oxygen available, leading to physical weakness and anemia. If left untreated, the anemia can impair mental function.
What is page?
PAGE, polyacrylamide gel electrophoresis, is a type of separation method in which polyacrylamide A polymer of acrylamide is used instead of agarose in the gel bed of the electrophoresis chamber. Polyacrylamide is preferable to agarose because it forms a tighter gel bed that is then capable of separating smaller molecules, especially those with molecular weights that are very similar. PAGE is used in DNA sequencing, DNA fingerprinting, and for protein separation.
What are GMO?
GMO are genetically modified organisms that have been created using new techniques of recombinant DNA technology. However, the term is misleading because almost all domesticated animals and crop plants have been genetically modified over thousands of years by human selection and crossbreeding. GMO are viewed with concern because of public debate over the safety of the products and the fear that GMO foods represent a type of biological pollution. The battle has been particularly intense in Europe, where GMO have been banned since 1998.
What feature of liverworts hints to their name?
Liverworts were named during the Middle Ages, when herbalists followed the theoretical approach known as the doctrine of signatures. The core philosophy of this perspective was that if a plant part resembled a part of the human body, it would be useful in treating ailments of that organ or part. The thallus of thalloid liverworts resembles a lobed liver. Therefore, in line with the philosophy presented by the doctrine, the plant was used to treat liver ailments. The word liver was combined with wort, which means herb, to form the name liverwort.
What is DNA sequence analysis?
DNA sequence analysis is part of a subfield of genomics known as structural genomics. DNA sequence analysis results in a complete description of a DNA molecule in terms of its order of nitrogen bases: adenine, thymine, guanine, cytosine. All sequencing projects are based on a similar technology: DNA fragmentation with restriction endonucleases. Cloning of the DNA fragments, and overlapping analysis of the fragments to eventually provide a map.
Who invented the thermometer?
The Greeks of Alexandria knew that air expanded as it was heated. Hero of Alexandria, 1st century CE, and Philo of Byzantium. Fluid ca 250 BCE, made simple thermoscopes, but they were not real thermometers. In 1592 Galileo, 1564–1642, made a kind of thermometer that also functioned as a barometer. And in 1612 his friend Sant'Orio 1561–1636, adapted the air thermometer, a device in which a colored liquid was driven down by the expansion of air, to measure the body's temperature change during illness and recovery. Still, it was not until 1713 that Daniel Fahrenheit 168611736 began developing a thermometer with a fixed scale. He worked out his scale from two fixed points: the melting point of ice and the heat of the healthy human body. He realized that the melting point of ice was a constant temperature, whereas the freezing point of water varied. Fahrenheit put his thermometer into a mixture of ice, water, and salt, which he marked off at 0 degrees. And, using this as a starting point, marked off melting ice at 32 degrees and blood heat at 96 degrees. In 1835 it was discovered that normal blood measured 98.6 degrees Fahrenheit. Sometimes Fahrenheit used spirit of wine as the liquid in the thermometer tube, but more often he used specially purified mercury. Later, the boiling point of water, 212 degrees Fahrenheit, became the upper fixed point of the thermometer.
How does a red blood cell carry oxygen?
Cells use oxygen to harvest the energy stored in molecules like sugars and fats. Red blood cells use a protein called hemoglobin to carry oxygen to all the cells of the body. Hemoglobin is actually made of 4 separate protein strands. Each one surrounds a central, iron-containing molecule. The hemoglobin group carries the oxygen molecule. Each molecule is made up of 2 atoms of oxygen bonded together.
Which aquatic animals have internal fertilization?
Certain sharks, skates, and rays have internal fertilization. The pelvic fins are specialized to pass sperm to the female. In most of these species the embryos develop internally and are born alive.
How does ergot affect humans and cattle?
Eating bread and other grain products contaminated with ergot causes the disease called St. Anthony's fire. Common during the Middle Ages, this disease, which causes sensations of intense heat followed by a complete loss of sensation in an infected person's limbs, no longer occurs very frequently due to improved techniques of grain production and milling. Cattle that graze on grains infected with ergot are able to ingest enough to cause death or the spontaneous abortion of fetuses.
What do the numbers inside the recycling symbol on plastic containers mean?
The Society of the Plastics Industry developed a voluntary coding system for plastic containers to assist recyclers in sorting plastic containers. The symbol is designed to be imprinted on the bottom of the plastic containers. The numerical code appears inside a three-sided triangular arrow. A guide to what the numbers mean is listed below. The most commonly recycled plastics are polyethylene terephthalate, PET, and high-density polyethylene, HDPE.
Who is considered the father of modern zoology?
Konrad Gessner, 1516–1565, a Swiss naturalist, is often credited as the father of modern zoology. Based on his three-volume Historia Animalium, which served as a standard reference work throughout Europe in the 16th and 17th centuries.
What type of signals control cell reproduction?
Cell reproduction is controlled by both external and internal signals. External signals include the availability of nutrients and space for growth. An example of an internal signal is the rise and fall of protein levels at specific points of the cell cycle, which is maintained by checkpoints and feedback controls. Cyclins and cyclin-dependent kinases are protein regulators that activate cell cycles and stimulate the synthesis of proteins.
Which bear lives in a tropical rainforest?
The Malayan sun bear, Ursus malayanus, is one of the rarest animals in the tropical forests of Sumatra, the Malay Peninsula, Borneo, Burma, Thailand, and southern China. The smallest bear species, with a length of 3.3 to 4.6 feet, 1 to 1.4 m, and weighing 60 to 143 pounds, 27 to 65 kilograms, it has a strong, stocky body. Against its black, short fur it has a characteristic orange-yellow colored crescent across its chest, which according to legend represents the rising sun. With powerful paws having long, curved claws to help it climb trees in the dense forests, it is an expert tree climber. The sun bear tears at tree bark to expose insects, larvae, and the nests of bees and termites. Fruit, coconut palms, and small rodents are also part of its diet. Sleeping and sunbathing during the day, it is active at night. Unusually shy and retiring, cautious, and intelligent. The sun bear is declining in population as its native forests are being destroyed.
What is an exon?
An exon is a segment of the transcript that is tied together with other exons by the spliceosome to make the mRNA molecule. After the exons are spliced together, they exit the nucleus for the cytoplasm, where the mRNA is translated into a polypeptide.
What is acid rain?
The term acid rain was coined by the British chemist Robert Angus Smith, 1817-1884, who in 1872 published Air and Rain, The Beginnings of a Chemical Climatology. Since then, acid rain has unfortunately become an increasingly used term for rain, snow, sleet, or other precipitation that has been polluted by acids such as sulfuric and nitric acids. When gasoline, coal, or oil are burned, their waste products of sulfur dioxide and nitrogen dioxide combine in complex chemical reactions with water vapor in clouds to form acids. The United States alone discharges 40 million metric tons of sulfur and nitrogen oxides into the atmosphere. This, combined with natural emissions of sulfur and nitrogen compounds, has resulted in severe ecological damage. Hundreds of lakes in North America, especially in northeastern Canada and the United States, and in Scandinavia, are so acidic that they cannot support fish life. Crops, forests, and building materials such as marble, limestone, sandstone, and bronze have been affected as well, but the extent is not as well documented as it is with fish life. However, in Europe, where many trees are stunted or have been killed, a new word, Waldsterben, forest death, has been coined to describe this phenomenon. In 1990, amendments to the U.S. Clean Air Act contained provisions to control emissions that cause acid rain. The amendments called for the reductions of sulfur dioxide emissions from 19 million tons to 9.1 million tons annually, and the reduction of industrial nitrogen oxide emissions from 6 to 4 million tons annually.
Who first suggested that apes could use language?
Samuel Pepys, 1633 to 1703, famous for his 17th century diary, wrote about what he called a baboon and suggested that it might be taught to speak or make signs.
What are the three major groups of chordates?
The chordates are divided into three subphyla: Tunicata, Cephalochordata, and Vertebrata. Tunicates are like little leathery bags that are either free-living or attached to pilings, rocks, and seaweeds. They are also called sea squirts because a disturbed animal may contract and shoot streams of water from both of its siphons. The subphylum Cephalochordata contains the Amphioxus or lancelet, branchiostoma, which looks like a small fish and has the three caudate features as an adult. Amphioxus also shows clear serial segmentation or metamerism. From the Greek terms meta, meaning between, among, after, and meros, meaning part, it is divided lengthwise into a series of muscle segments. Vertebrates, which comprise the third caudate subphylum, retain the same metamerism in internal structures.
What is zero population growth?
Zero population growth, or ZPG, is the estimation of the birth rate necessary to maintain the size of the human population at its current level. As of now, the rate is estimated as 2.1, which means that each set of existing parents would need to have, on average, slightly more than 2 children during their lifetime. The extra 0.1 allows for infant mortality.
What traits of peas did Mendel study?
Mendel studied peas of distinct and recognizable plant varieties. Mendel's experiments included the characteristics of height, flower color, pea color, pea shape, pod color, and the position of flowers on the stem.
What is limnology?
Limnology is the study of freshwater ecosystems, especially lakes, ponds, and streams. These ecosystems are more fragile than marine environments since they are subject to greater extremes in temperature. The study of limnology includes the chemistry, physics, and biology of these bodies of water. F. A. Farrell, 1848 to 1931, a Swiss professor, has been called the father of limnology.
How does an independent variable differ from a dependent variable?
An independent variable is manipulated and controlled by the researcher. A dependent variable is the variable that the researcher watches and/or measures. It is called a dependent variable because it depends upon and is affected by the independent variable.
What is the largest jellyfish?
The largest jellyfish is the genus Cyanea capillata. It may be more than 6.5 feet 2 meters in diameter and have tentacles of 98 feet 30 m long and is among the largest invertebrates.